Hello and welcome to this module on RNA secondary structure motifs. This will only be a brief introduction to RNA motifs. I will be following up this module with another discussion on the energetics involved in secondary structure formation within RNA, as well as the prediction algorithms that can be applied to predict secondary RNA structures. So let me begin by highlighting the differences between RNA and DNA. And I want to begin by discussing the commonly remarked differences that we understand between RNA and DNA. Some of these differences, or the major differences, are that RNA is single-stranded, whereas DNA is double-stranded. Of course, based on the central dogma of biology, we understand that DNA will produce RNA via transcription. There are different types of RNA, including messenger RNA and non-coding RNA, which has various other subtypes. And we think of RNA as a single-stranded sequence, whereas DNA as a double-stranded sequence. Now, this is a valid way to look at RNA, but in reality, RNA can form secondary structures. And this is the case not only with non-coding RNA, where these secondary structures are vital for the non-coding RNA's various different functions, whether they are housekeeping or regulatory functions. And it's also equally important in messenger RNA, where the formation of certain secondary structures will help regulate gene expression. It is quite the case that we don't think of messenger RNA in particular as being a strand of RNA that forms secondary structure, because we always think of a messenger RNA sequence that will be bound by ribosomes and undergo translation to produce a final protein product. But in reality, even messenger RNA can form secondary RNA structures that will help regulate gene expression. This will not be the topic of this discussion. That is the discussion of the secondary structures in messenger RNA and how that will help regulate gene expression. But rather, I will be discussing the common motifs or the common secondary structures of RNA in general. Another common difference that we understand between RNA and DNA pertains to nitrogenous bases. For example, we understand that in RNA, we have uracils as opposed to thymine that is found in DNA. Now, it's difficult to know why RNA uses uracil as opposed to thymine, and there is no conclusive theory that sufficiently explores this topic. Yet, as with most things, the answer is pertinent to evolution and cost-benefit analysis of using uracil in RNA as opposed to thymine that we see in DNA. And lastly, a third difference that we understand between RNA and DNA is pertaining to the ribose sugar. Specifically, if you look at the 2' carbon, this right here is the 2' carbon. In DNA, the 2' carbon does not have a hydroxyl group attached to it. So here you can see only 2 hydrogen atoms that are attached to the 2' carbon. And thus, in DNA, we find deoxyriboses. On the other hand, in the RNA sequence, we will find riboses as opposed to deoxyriboses. That's because if you take a look at the 2' carbon, you will find that it is attached to a hydroxyl group, and thus it does not have a missing hydroxyl group, and thus it is simply called a ribose sugar in the nucleotide triphosphates of RNA as opposed to a deoxyribose. Importantly, before I get into the secondary structures of RNA, I want to briefly discuss the types of RNAs that we find. A fundamental type of RNA that we understand all too well is the messenger RNA. And obviously, the messenger RNA is the RNA that will be utilized as a template for translation and protein synthesis. On the other hand, we have another category of RNA, which has a number of different RNA types under this category, and that is the non-coding RNA. By definition, non-coding RNA do not code for the proteins that we generate in our cells, and thus these RNAs do not code for any protein synthesis. And the types of non-coding RNAs include transfer RNAs, ribosomal RNAs, as well as long non-coding RNAs, such as the telomerase RNA, as well as short non-coding RNAs. Now, as I said earlier, secondary structures will form in all types of RNAs. This includes even short non-coding RNA, as well as the messenger RNA in certain regions, which helps regulate gene expression. But of course, secondary RNA structures also play a pivotal role in tRNA, ribosomal RNA, and long non-coding RNA. And the secondary structures that I will be discussing in this module can be found in most types of RNA. So once again, I do want to stress the fact that RNA is not merely a single-stranded RNA sequence. It is the case in messenger RNA in most regions that will be translated, but even in messenger RNA, you'll have secondary structure formation to regulate gene expression. In the different types of RNA, regardless of type, you can have the formation of secondary structures. This is of particular importance in tRNA, ribosomal RNA, as well as the long and short non-coding RNAs. The secondary structure is basically where a sequence of RNA will fold on itself. So you can have base pairing between this region of the RNA as well as this region of the RNA. This base pairing can form different motifs, and then this base pairing can be more complex. For example, you can have this region, then base pair with this region. So the major idea that I want to bring across is that secondary structures can form, and we call the various commonly found secondary structures as motifs, and we label them with various names, and then we study their stability and energetics, as well as various algorithms that we can use to predict their formation. And different motifs will have different ways of having them predicted. That is, if you have a particular RNA sequence, you can predict different motifs with different models and algorithmic principles that can help predict the overall structure, the overall secondary structure, that is. So for example, if we see a pattern of secondary structures such as this one, we can label it with a term that can assist us in scientific communication of that term and further study of this particular motif. So what I circled, for instance, is called a hairpin loop. It's basically where you have a loop or a group of nucleotides that's closed off by a canonical base pair. I'll discuss what a canonical base pair is later in the slides, and also discuss the hairpin loop in greater detail further on. You can also look here at what I'm circling. These are called helices, and they consist of canonically paired nucleotides. This right here would be a dangling end, and on, as you will see in the next slides. Importantly, after the formation of the secondary structure, you'll also have the formation of a tertiary structure. The secondary structure is basically a structure in 2D. It's sitting on a plane, on a singular plane, that is. And energetically, what will be more favorable and what will actually happen is that the different parts of the RNA secondary structure will bend and twist onto one another to form the most stable possible structure. So for example, you can have this portion of the secondary RNA structure bend this way in a particular way in a 3D multiplanar scenario. And what you will form as a result of that is a tertiary structure. This is a structure in 3D where you have various bending of the secondary structure components of the RNA. Importantly, the major discussion here is the secondary structures of RNA, as well as the different labels that we ascribe for the various common motifs or particular secondary structures that we see in RNA. So let me begin by introducing the first secondary structure, and the simplest and the most fundamental secondary structure in RNA, and that is the helix. So important to understanding helices is understanding canonical base pairing. So canonical base pairing is where you have a guanine base paired with cytosine, or an adenine base paired with uracil, or a guanine base pair with uracil. Importantly, you can notice here that canonical base pairing does not simply refer to the Watson-Crick base pairing. For example, we understand that guanine and cytosine here are Watson-Crick base pairs, as well as adenine and uracil. They're also Watson-Crick base pairs that we would expect in a secondary structure of RNA. However, the guanine uracil is a wobble base pair. It's still a canonical base pair. It's not a Watson-Crick base pair, but however, it is also a stable base pair that can form. The other wobble base pairs, there are basically four main wobble base pairs. They include inosine with cytosine, as well as inosine with uracil and inosine with adenine. And the fourth one is guanine with uracil. It is only the guanine uracil base pair that is considered a canonical base pair. It's a relatively stable base pair, not as stable as the Watson-Crick base pairs, but it's still pretty stable, and it is a canonical base pair that you should consider. So a helix, which is the singular term of helices, is a group of two consecutive canonical pairs. So basically, anytime we see two consecutive canonical base pairs or more, then we call these as helices. This is a type of an RNA motif, and it is a fundamental RNA motif that you should be familiar with. For example, here you can see helices. The helices are composed of these base pairs that you can see here. Specifically, these are canonical base pairs. So for example, you might have a guanine here and a cytosine here, or you can have an adenine and a uracil, or you can have a guanine and a uracil, which is also a canonical base pair, despite not being a Watson-Crick base pair. You can also see helices here, as well as here, and here, as well as here. Another type of an RNA motif is the dangling end. So a dangling end is an unpaired nucleotide or region of nucleotides at one end of an RNA structure. For example, if you take a look here, you can see that you have helices present here, but at the very end, you have a nucleotide that is unpaired, meaning that there is no base pairing with another nucleotide or another nitrogenous base in this strand. Now, particularly a 3' dangling nucleotide has a similar stability to a non-canonical base pair in a terminal position, meaning that if we assume that this right here is a 3' dangling end, then this 3' dangling end would have the same stability as if those base pairs, those terminal base pairing that we see here, is if it was a non-canonical base pair. Now, if this was a canonical base pair, then the 3' dangling end would not have as much stability as a canonical terminal base pair. So dangling ends can stabilize a helix indeed, but it is not as stable as a terminal canonical base pair. Now, before I move on, I do want to clarify some things. So I said that if you were to imagine these as non-canonical base pairs, here I'm talking about these two nucleotides at the very end, you want to keep in mind that these are in fact terminal mismatches in the figure. So meaning these do not base pair with one another. So before we move on, I do want to clarify some things. So if we look at the figure here, which you'll see across the slides that are upcoming, you will notice that this looks as if you have two strands of RNA, meaning it looks as if you have a strand that starts this way and continues on and on, as well as a strand that starts this way and continues. But in reality, this is merely a single strand that will create some shapes and eventually it loops around. So that's what I want you to keep in mind. So if you were to imagine that this 3' end is right here, you have to keep in mind that the 5' end is actually right here. Assuming that this indeed is a 3' end, which is most likely is, considering the 3' dangling ends are more stable. So the point that I want to make here is that these right here are not in fact base paired with one another. And further on that point, when you have the RNA twist in a stark turn, as you see here, assume you have one nucleotide here and one nucleotide here, we do not expect those nucleotide to base pair with one another. So, for example, if you were to go through a particular algorithm that is supposed to predict secondary structures of RNA, inherent to that algorithm is that you do not, or you cannot have base pairing at a stark turn that includes the very nucleotides that are involved at the highly angled turn, if you will. So please keep this concept in mind, and also keep the concept that what we're looking at here is simply a single-stranded RNA that has twisted on itself to appear as if it is double-stranded RNA. So in reality here, this base pair, or excuse me, this nucleotide that you see here and this nucleotide that you see here are connected with one another, just like that. So for the rest of the figure, please just imagine that those nucleotides are indeed connected at the end. The rest of the other RNA motifs that I'm going to discuss are actually all loops. So you can imagine what a loop would look like. It's basically where you have an RNA sequence that is forming some kind of a loop. The RNA motifs that have loop shapes include the hairpin loops, the bulge loops, the internal loops, the multi-branch loops, which are also called junctions, and the pseudoknots. The pseudoknot is probably the most complicated motif loop, and I will discuss that at the very end. But in the figure to the right, you can see a hairpin loop here. I'll discuss the exact features of a hairpin loop in a second. You can also see the bulge loop here to the left. There is also the internal loop that can be seen here, as well as the multi-branch loop and the pseudoknot, which is not present in this figure. Now, looking at these loops, you might see them as all being very similar and the same, but there are differences in all of these different loops that I highlighted. Each one of them is characteristic of particular features, which I'll discuss in the next slides. So let me begin by discussing the hairpin loop. So a hairpin loop, by definition, consists of several nucleotides that are unpaired. And importantly here, you have to recognize that these nucleotides are unpaired, entirely unpaired, and closed by a single canonical base pair. Here to the figure on the right, you can see a hairpin loop. You can see that all the nucleotides that form this loop are unpaired, meaning you don't have a base pair here, for example, between those two bases or those two bases. And importantly, as part of the hairpin loop, this loop is closed off by a canonical base pair. This is the canonical base pair that will close off this hairpin loop. Another type of an RNA motif that is a loop is the bulge loop. A bulge loop, by definition, is an obtrusion or an interruption of helices through unpaired nucleotides in one strand. Now, this is a very particular definition. So you have an obtrusion or an interruption of helices. We've already defined helices. They consist of two consecutive canonical base pairs, and the obtrusion happens because of unpaired nucleotides in one strand. So you have to keep in mind the fact that this is caused by an obtrusion of helices, and also you have to keep in mind that the obtrusion happens through unpaired nucleotides in only one strand. So to the figure on the right, you can see a bulge loop here on the left. You can see that you have a loop structure. And you can also see an obtrusion in the helices. The obtrusion is what is causing the loop. So the obtrusion is caused by unpaired nucleotides in one strand. So what do I mean by that? If you take a look at this sequence of RNA, you can notice that you have base pairing in all of the nucleotides within the sequence. For example, in this nucleotide within that sequence that I just highlighted, you can see that there is a base pair. So this nucleotide is part of the bulge loop and it is forming a base pair. In addition, if you look at this nucleotide, it also engages in base pairing and it is part of the bulge loop. However, what you have is those two nucleotides which have nitrogenous bases that are unpaired. And these nitrogenous bases that are unpaired are only found in one strand. They are found in this strand of the loop. And these are the nucleotides that are interrupting the helix. This is the helices here that you can take a look at. And these helices that you see are obstructed or interrupted, or there is an obtrusion of this helices RNA secondary structure motif caused by these unpaired nucleotides within one strand. So it's very important to understand that in bulge loops, the interruption of the helices is caused by unpairing of nucleotides within only one strand, meaning that the nucleotides within this strand are completely paired. This is in contrast with an internal loop. So an internal loop can be seen here to the right of the figure. And an internal loop, by definition, is an obtrusion or an interruption of helices through unpaired nucleotides on both strands. So if you take a look here at this internal loop, you'll notice that in the internal loop, you will have unpaired nucleotides on this strand. So in the yellow, you can see that there are two nucleotides that have no base pairing, as well as the unpaired nucleotides in this strand of the loop. You can see that these two nucleotides also do not have any base pairing. So you can see that in an internal loop, in contrast to a bulge loop, you have an interruption of the helices that is caused by unpaired nucleotides on both strands and not just one. So this is the overall helices that you can see here, the RNA motif helices, and they are obstructed by this internal loop because you have unpaired nucleotides on both strands of this RNA helices. Now, alternatively, the internal loop could be an interruption of helices through non-canonically paired nucleotides on both strands. So I don't have a figure that showcases this, but suppose that you have a helix structure right here. You can see here this is a helix structure, and let's just assume that you have base pairing all throughout these parts of the helices, with the exception of this region right here where you have a loop. Now, you can imagine that this loop, for example, is an internal loop with, say, two nucleotides here and two nucleotides here. I know this is a bad-looking loop, but just bear with me. And these two nucleotides could be completely unpaired, but on the other hand, this would also be considered an internal loop if those nucleotides do indeed base pair, but that this base pairing is between non-canonical nucleotides or non-canonical base pairing. So, for example, if this nucleotide that you see here and this nucleotide engage in base pairing that is non-canonical, non-canonical meaning that it is any base pair that is not a UA or GC or GU. If it is any other base pair, it would be a non-canonical base pairing. And if that is the case, then those base pairs, if they are non-canonical, they could cause an internal loop to be formed. Another type of RNA motif that is a loop is the multi-branch loop. This is also called a junction. A multi-branch loop is also called a junction, and by definition, a multi-branch loop diverges into 3 or more helices. So they do have to be 3 or more helices. And here to the figure on the right, you can see that you have a multi-branch loop. This is a loop. Obviously, you can see it's a loop and it diverges into at least 3 helices. So you can see that you have a helix that diverges to the left. You can see it right here. You can see that you have one that diverges to the top here and one that diverges to the right. So by definition, this is called a multi-branch loop or a junction. And lastly, I want to discuss the last RNA motif in this module, which is the pseudoknot. So before I introduce a formal definition of a pseudoknot, I want to introduce a particular type of a pseudoknot, which is the most characterized type and the easiest one to understand, and that is the H-type pseudoknot. So pseudoknots come in 4 major types. They're called the H, K, L, and M types. The H is the most well characterized and is the simplest to understand. And if you understand the H pseudoknot, then it's easier to understand the K, L, and M pseudoknots. But for now, I want you to understand the H-type pseudoknot. An H-type pseudoknot is formed when bases within a hairpin loop base pair with nucleotides outside the hairpin loop. So recall that a hairpin loop, by definition, is where you have a group of nucleotides that are completely unpaired and that are closed off by a canonical base pair. So here you can see, for example, a helix that I am drawing, and on the top of this helix is a hairpin loop. This hairpin loop is closed off by this canonical base pair that I'm circling, and this right here would be a hairpin loop. The nucleotides within the hairpin loop do not base pair with one another, and they are closed off by a canonical base pair. So the idea with an H-type pseudoknot is that you have a hairpin loop, and the bases within the hairpin loop, that is inside the hairpin loop, will base pair with nucleotides outside the hairpin loop. I'll show you what this looks like in a second. This right here is a figure that discusses the various types, the 4 major types of pseudoknots. And I want to discuss the H-type here, mainly by evaluating the figure that you see at the bottom. So at the bottom, you can see a linear RNA sequence. Here is the RNA sequence. And what I want you to notice here is that you have some bases, some nucleotides in this region. These will base pair with nucleotides in this region. And you have some bases in this region of the RNA linear strand, and they will base pair with this region. In reality, you can think of this as first the formation of a hairpin loop. So here to the bottom of the figure, you can see that you have a hairpin loop. It consists of nucleotides that are completely unbase paired or have no base pairing between them that are closed off by a canonical base pair. And so this hairpin loop, you can take a look at how it was formed. As you can imagine, it was formed when this region of nucleotides base paired with this region of nucleotides. Mainly when this base pairing happened, this formed the hairpin loop. And you can see this by the color-coded figure. So here in this hairpin loop, you can see this region of the RNA linear stranded sequence. Here in the stem of the hairpin loop where you have the helix, you can see that you have the red sequence or red region of the RNA sequence, as well as the green region of the RNA sequence. So basically, when this structure has undergone base pairing between this region and this region, this formed the stem that you see here, and it also formed the hairpin loop. Now, once the hairpin loop is formed, the bases or the nucleotides within this hairpin loop can base pair with a region of the RNA sequence that is not part of the hairpin loop. So you can see here that you have some base pairing with this blue part here at the bottom at the 3' end of the RNA strand. And this base pairing is what will form the overall pseudoknot, the H-type pseudoknot that you can see here to the bottom right. Now, I can appreciate that it can be complicated to understand how the structure here to the left was transformed into the one to the right. For example, in the one to the right, you can see that you have a loop in here to the left. This is a loop where you have no base pairing, and you can see that you have a stem. The stem refers to those two regions that are base pairing. It's a helix. You can see that you have another stem. This is the stem, as well as another loop, which refers to this circular structure here. So to make it simple, I'll actually do some drawing here. So if you take a look at my pen to the bottom left, I'll do my best to do some drawing that showcases how we go from here into here, into the C part of the figure. So if we can imagine that we are at B, so let's draw B here. We have the 5' end here, and this 5' end has a hairpin loop at the end of it, and this hairpin loop will also extend this way off of the helix, and you'll have the 3' end. So obviously here you have a non-canonical base pair that closes off the hairpin loop, but the idea here is that you have some nucleotides that are going to base pair with the region at the 3' end. Now, importantly, to understand how we go from here to the very end at the bottom right of the bottom figure, we have to draw some simpler steps in between B and C. So let's do that. So to make it easier, let me draw an intermediate step that happens before. What I want to draw is the actual base pairing, what happens when this region right here becomes closely associated with this region in the hairpin loop. So let's draw that. So we have the 5' end here, and then we have what used to be the hairpin loop, and then we have the 3' end. But now the 3' end, or a region within the 3' end, has gone up and has base paired. This is the 3' end. We transferred it from here into here, and it is base pairing with a region within the hairpin loop. So to understand what happens next, I'm basically going to redraw this, but this way rotating it to the right in a vertical position. So if we redraw the structure with the 5' end here at the bottom, and then we draw the following structure, I'm going to draw the loop that you see here. So if we draw this loop as looking like this, and then we have this structure like this that goes down, and then now I'm getting to the 3' end. All I did here was that I just rotated, as I told you here, this particular region where you have the base pairing. And now you have the base pairing looking like this, and you have the stem that we had originally right here. So what you notice here is that now you have this loop. This right here is a loop. This region is basically a loop. Here you have the new base pairing. This is a stem. And then you also have the original stem that we had, as well as another loop. And this is the exact same structure that you see in C of the bottom right figure. Now that we've introduced the H-type pseudoknot and have a general idea of this particular pseudoknot, I do want to introduce a general pseudoknot definition. So this is more or less a universal pseudoknot definition. It's a way of defining pseudoknots in an algorithmic way, which can be very helpful. So this pseudoknot definition says that a pseudoknot is basically where you have at least two base pairs. These base pairs we'll call IJ base pairs, and I prime to J prime base pairs, where you have the I region of the RNA sequence come before the I prime, which comes before the J region, which comes before the J prime. So I and J are utilized as nomenclature that can signify either a singular nucleotide or a region with several nucleotides. And so you can think of I as a region of nucleotides that base pairs with a region of nucleotides, which you can think of or call J. And then you can think of I prime as a region of nucleotides that can base pair with another region of nucleotides, J prime, and this is obviously within one singular RNA sequence. So, for example, if you take a look to the figure at the bottom right and we take a look at A, we can consider this region in red as I, and then we can consider this region in yellow as I prime. We can then consider this region in green as J, and then we can consider this region in blue as J prime. So in this definition, I must come before I prime, which must come before J, and must come before J prime. And then you simply follow what the definition says. So you have base pairing between I and J. So you can see here that I and J are base pairing. And then you have base pairing between I prime and J prime. So we can apply this definition to the figure that we see in the top. If we call this as an I region, and we call this as an I prime region, and we call this as a J region and this as a J prime, notice that the I will base pair with the J region, and then the I prime will base pair with the J prime region. And you have the formation of an H-type pseudoknot where you have a hairpin loop that's going to form, and this hairpin loop will have nucleotides within it that will base pair with a region outside of the hairpin loop. So once you understand the H-type, it's pretty intuitive to understand all the other types of pseudoknots. And the way I recommend that you understand them is basically by seeing the H-type pseudoknot and then thinking of additions or more complexities that will be added to the H-type pseudoknot in the other types of pseudoknots. So for example, if you're looking at the K-type pseudoknot, I want you to focus at this base pairing that you see here. You can think of this as being an H-type pseudoknot with the exception that you have an additional complexity caused by the base pairing that you see with this half circle here. So you have an H-type pseudoknot that you can see here in this K-type pseudoknot. And the reason it's a K-type pseudoknot is because you have an additional complexity that you see here. And this additional complexity is what's causing this to be a K-type pseudoknot. Similarly, you can do this with the L-type pseudoknot. Again, you can think of this structure here, more specifically the base pairing that you see here, as being an H-type pseudoknot, with the difference being that you have this additional complexity. The difference here between this complexity and the one that I noted in the K-type pseudoknot is that the base pairing that happens here is such that there is interconversion or intersection at the point right here. And lastly, you can do the same thing for the M-type pseudoknot. You can imagine that this right here is your H-type pseudoknot with some complications that are added here so that you have an M-type pseudoknot. And this concludes this module on RNA secondary structures and motifs.